Hello, my name is Mr. McQuire, and today we are going to be studying the lofty topic of monoclonal antibodies. So this lesson is a biology lesson for triple science, and it's a higher tier only. I'm going to split the lesson into two because we're going to be looking later in the second video an aspect of plant biology that fits at this point as well. You will need the following equipment: a pen, paper to record and to revise, and you will find that there is a Google document to accompany this lesson. Once you've found this equipment, unpause the video and then we can start work. So this is the syllabus for the lesson that we're looking at today. If you look in detail at it, you can see that we're going to look at the production of monoclonal antibodies. We're also going to be looking at the uses, but that includes, down at the bottom, some ethical issues that may arise that we need to be familiar with as well, that fit here. There are some very important keywords that we're going to get used to using, and we're going to revise them. So you're going to need your piece of paper, which could be scrap paper, or you can record these on the Google document if you've got direct access to it because you printed it out, for example, or you can flick between the two. So here's our pre-starter. I'm going to give you a couple of minutes on this and let's see how you do. We'll run through some words in a moment and look at some associations. So starting from the top, what does the prefix or the word at the beginning mono mean? What does the word antibody mean to you? What does the word clone mean to you? And finally, a bit of a funny question: what is a hybrid car? So I'm going to give you 2 minutes on that. Now I really want to use your minds to help you remember these expressions, because they're going to be really critical for these higher level answers that we need in biology on a higher paper. So I've put together some thoughts that I'd like you to just smile at, allow to move into your head, to remember, and I've looked for things that will help you link with them. So what does the prefix mono mean? Now this is a very famous model. She has majored on the fact that she has a monobrow, and she's not ashamed to show it, as you can see. So the word mono means one, of course. Here's an antibody. This is a picture of an antibody that belongs to the flu virus. Maybe that's the association that you have in your head. I'd like you to remember that kind of picture as we run through. Now, what about clone? Do you have a mental picture of what clone means to you? For some people, of course, the process of cloning might result in identical animals, and it's certainly the case that people spend a lot of money having dogs cloned these days. Maybe, like me, you're a little bit older, younger, whatever, and maybe clone has something to do with these characters. Hybrid car, of course, is a different question. Maybe you're not a car nut, but maybe you understand that a hybrid car is one that has both a petrol motor and an electric motor, and hybrid means that it works between the two. And you're going to see that word is very, very important as we work through this particular topic. So this is a section on monoclonal antibodies, and we're producing monoclonal antibodies in a response in our body that's very, very specific these days. This is high-end medicine. I have to say that it's not really met the expectations that a lot of doctors had, and we're going to look at some of the issues that have occurred that means that this, which was meant as the saviour of lots of very sick people, really hasn't lived up quite to its expectations, but is beginning to do so now. So here's some revision. You'll need your paper and you'll need your pen, and I'm going to help you revise some words. I'm going to give you a couple of minutes to allow us to work through this particular sheet together. In front of you we have some of the most important words, and I'm going to suggest to you that we're going to record them either on a piece of paper that you've got or your Google document, because we need to learn them. We're then going to spend some time just practicing with those words, and we're going to do that as you would maybe with something called look, say, write, cover and check, and I'm going to help you to do that as we work through this. We can't write a story of the production of monoclonal antibodies without using these words, so let's learn those words now. So as you can see on the document that's straight in front of you, what is a clone, what is an antigen, and how are antibodies specific? On the right hand side of the screen you can see that's the top document that I've just enlarged, but there's also a second document on the right with another set of questions. So we've got these 6 questions to learn the answers to. So initially what I'd like you to do, please, is to just record the information that is in front of you. If you've got the Google document open, then I need you to think hard. Hopefully the environment where you are is quiet and you've got an opportunity to actually fill these in on the Google document, answering those particular questions. You can literally copy the answers down. I'm going to give you a good 10 minutes to enable you to do that, and then we're going to do some revision of that information. Remember, we're recording the information that's in front of you at the moment. If you've completed writing it down, start looking at the information that you've got. Remember that we're trying to associate, for example, the top box, what is a close. If the principal word in that response is the word identity. Let's try revising so that we associate the word clone with the word identity. Are there any other words in that box that you would use? Perhaps if you've got a highlight, you could highlight them. given a great chunk of time to the recording of that information, and very purposefully, just like speaking French, it's critical that we use those words when we're answering questions. This work is really high-level work, and it demands that we use those words appropriately while we're trying to answer them. So let's see how we can structure some revision. If you've got the work in front of you now and you've recorded it, if I covered up that side and asked you to fill in the gaps that were there, do we remember what the key words are, what the important words are that we were trying to remember? So the first task I'd like you to do is to have a go at being able to recall what the principal words are that are underlined on that right hand box. Now you can pause the video at any point to allow you to practice these, but it would be great if we got to a point where we understood those and knew the responses. In a short while I'm going to move the cover that's currently on the right hand side across to the left hand side, and I'm going to ask you what we're describing, because we need to know these responses in both directions. Okay, so I'm not going to run this in silence now, because I need you to be able to run this through and revise it to a point where you are fluent in knowing what the word clone and associating with identical, with the word antigen, and the knowledge that it's connected with a binding site on the surface of a pathogen. Indeed, what the word pathogen means, but also antibodies, which are connected very strongly with that word one. Very specific. Antibodies are specific. What does that mean? So I'm going to cover this up, pause it, practice, okay, and then I will come back to you in a moment. Okay, I hope you're now familiar with what's underneath there and you've practiced those. Now I'm going to move it to the other side, if you can repeat the same process so you know exactly what the key words are from the definitions. Okay, we're going to move on now. So hopefully you've now got an understanding of those three words and what the definitions are. So here's the other sheet. Now we can repeat exactly the same process, but I'd like you to work unassisted for the next few minutes. There's space on the Google Sheet for you to do that, or hopefully you have some paper that you can record that information on if you can't see the Google Sheet and work simultaneously with that. This really is critical information, and it will take you a good 10, maybe 15 minutes to be able to record that information. Okay, we're going to again revise that. So I'll cover that side up. Please pause it. Look at the keywords and associate it with the word that's on the left hand side. Let's look at that again. So a lymphocyte, a type of white blood cell that makes antibodies, but it's not capable of dividing. These are critical expressions that we're going to be using. So I'll cover that up and just pause it, and let's do some written revision. I'd be scribbling that with the keywords and just making sure I understood that. Now moving it to the other side, let's reverse the association. So I know the top one is lymphocyte. Okay, you can see that I chose some keywords at the start of this particular topic. The word clone is in one of the words we're trying to learn as well. We've got antibodies there, and you can see this expression hybridoma down at the bottom that we're going to see more of. So it's quite controversial science that we're moving on to now. It's important that you have an ethical standpoint on whether you agree with this particular science or not, but you need a reason. You need to be able to express that reason in the event that you're asked about it in an exam. Let's go into the technicalities though of how monoclonal antibodies are produced, as you can see in the Aspire, and then some ways that monoclonal antibodies are used, and we'll look at the ethical issues. You can see that in the challenge above. Okay. This is a brilliant diagram, and this is an essay plan in itself. It's really important that we can go through each of the stages that are here. So the step one I want to point out to you is that there are 6 stages on the diagram above. You can see that there are some key words in here. Step 1 has the word immunisation. Step 2 has the word isolate. In step 3 we have the word fusion, the joining together. Step 4 we're screening. Step 5 we've got the word cloning. And then finally the monoclonal antibodies, that's the whole production process point. So those are the critical stages that we need. If we were writing this in the list, they're the words that we need to understand. The point of this whole area of science is to produce a personalised antibody for the treatment of a singular person who has an infection that their body is not dealing with. It's really critical that you understand that. This is why this was seen as the future of science. A person who has an infection and that person is seriously ill with it and cannot fight it, they discovered that they could begin to use a process that brought together different parts of science to produce specific antibodies that would work for just that one individual. So starting with the first process, the first thing we do is we collect the copy of the pathogen from the individual who's ill. In step 1, the mouse in this case is immunised with that infection. The mouse has an immune response in step 2 and forms antibodies to that particular infection that's come from the human. Now we need to get hold of those antibodies, and at that point there, for those of you with more vivid imaginations, you can imagine the role of the mouse at that point in this particular operation. This is a clever part now coming to step 3. If you look across, we need to find a way of replicating this immune response. So what scientists did was they found cells that are incredibly good at replicating, which is a tumour cell from a cancer, and they removed the nucleus from the tumour cell and they replaced that particular nucleus with the information coming from the mouse immune response. So we've now created a hybrid, literally, of a mouse and a human cancer cell. You can sense the controversy or the argument that's coming into this science, and perhaps begin to understand why we have some issues with rejection and with side effects to this. Once we've created this hybridoma cell, and it's critical that you learn that particular word, what is it a hybrid of? A tumour cell and mouse antibodies. Then at that point then we allow it to reproduce by cloning. That's asexual reproduction, and on here that's step 5. And at that point then we're farming those desired antibodies that are capable of fighting the infection. Now once we're at step 6, those antibodies then, which are now specific to the human with the infection because they're part of their DNA, can then be inoculated, injected back into the patient that they came from. So this patient is being treated with a specific antibody that will only work for their particular infection. So these are very rare infections that we're treating. If you need to hear that description again, then please rewind the video to allow you to do that. So I'll give you time now just to answer that particular piece of work. So we are looking for what is the production of monoclonal antibodies, and we are writing a response onto the 6 sections and keywords that are on the Google Sheet. Rewind my description if you need further help with each of the stages. Okay, by this stage we should have some keywords that we have learnt right at the beginning of the lesson. Take your mind back to somebody with a single eyebrow and to pictures of Star Wars characters. We've also begun to get a sense of the fact that there's some ethical issues involved here because we're mixing human cells with animal cells, and there are implications for the happy life of that mouse that you can see in front of you. But we are producing some amazing personalized monoclonal single cloned antibodies to treat some very sick people with those on the other side of that. If you need more time to record the process, look at the Google Sheet. It has a structure to allow you to do that work. Now, moving on to the challenge, we need to know some quite recallable information now. There's 1 or 2 bits I want to point out to you as we... Okay, just to recap what you should have in front of you now. You should have a list of particular terms and their definitions, and there should be 2 blocks of those that you've revised. You should also have a description of the process of the production of monoclonal antibodies. You should then have a piece of work which is the challenge in front of you, which is to describe monoclonal antibodies use, but also to start the process of looking at ethical issues, advantages and disadvantages, side effects that come with that, treatment using animals, the mixture of human and mouse cells, etc. that run with this. So you should have a broader knowledge now of that particular information. So here we are at the plenary. All I would like you to do is to, on your Google Sheet, just to complete the following sentences. It'll only take a couple of minutes to do that. So a hybridoma cell is a combination of what and what. Please notice the word hybrid because that should be stuck in your mind now. A use of this technology is, and you should have an example that sticks in your mind. I've told you my example that sticks. Some people think the use of monoclonal antibodies is unethical because, and again, just have an opinion that's very clear that you can put into that. This is quite a straightforward answer or question, but sometimes this is the depth that we go to rather than having full 6-mark descriptions of the production of monoclonal antibodies. Now I'm going to give you a full-timed couple of minutes before we just recap at the end of this lesson. recording the gaps or filling the gaps in on your Google Sheet. Okay, I hope that you've learned a lot about monoclonal antibodies today. They are cutting edge bits of science, and it has some very, very important keywords that go with this high level of science that's used now in the National Health Service on a daily basis with some very, very poorly people. As we know, it's very expensive, it's controversial, but it's keeping some children, especially with some very serious previously untreatable illnesses, it's giving them now completely recovery, and that's still raising headlines that you will see in the newspapers today. However, there are ethical issues that you need to be aware about. Now, on the document in front of you, it's already linking through into the next part of the lesson, which is connected with plants. The Aspire there describe plant disease and how they are detected and how plants defend themselves from diseases, and we'll be looking at that in the next part of the video. Thank you for staying with me on this one.